Embryology is a branch of biology that studies the early development of organisms from fertilization to the formation of more complex body structures. Before organisms reach the stage of birth or birth in mammals. In humans, embryology focuses on the stages of fetal development in the womb. Starting from fertilization of the egg to the stage of birth. Understanding embryology is very important in the medical field, because it provides insight into the origins of human development and disorders or diseases that can arise during the process. The following is a detailed explanation of embryology. Starting from the basic processes and stages of embryo development, to its clinical applications. 1. Fertilization process. Fertilization is the process of meeting between the egg, ovum, from the mother and the sperm from the father, which produces a zygote. This process occurs in the fallopian tube, uterine tube, and is the first step in human embryology. Fertilization steps. 1. Sperm penetration. Sperm that enters the vagina moves through the cervix to the fallopian tube to meet the egg. 2. Fusion of sperm and egg cells. Sperm that successfully penetrate the protective layer of the egg cell, zona pellucida, will fuse with the egg cell membrane, which triggers fertilization. 3. Formation of zygote. After the sperm enters the egg cell, the genetic material from the sperm and egg cells combine to form a zygote, which is a single cell that has a complete number of chromosomes, 46 chromosomes in humans. Results of fertilization. The zygote is a diploid cell, has 2 sets of chromosomes. 1 from the mother and 1 from the father. This zygote begins to divide and develop into a more complex structure in a process called cloning. 2. Stages of embryo development. After the formation of the zygote, the human embryo undergoes several highly organized stages of development. These stages include cell division, cell differentiation, and the formation of important organs. These stages can be divided into several main phases. A. Zygote cleavage. In the early stages after fertilization, the zygote will undergo rapid cell division. This division occurs without cell growth, so that the overall size of the embryo remains the same even though the number of cells continues to increase. Morula. After several divisions, the zygote turns into a morula, which is a solid ball consisting of a number of cells called blastomeres. The morula is formed about 3 to 4 days after fertilization. Blastula. The morula develops into a blastula, where the cells begin to form a cavity in the middle, blastocele. This blastula will eventually become an important structure for implantation in the uterus. B. Implantation. About 5 to 7 days after fertilization, the blastula will attach and enter the uterine wall, endometrium, in a process called implantation. This process is important to provide nutrients and oxygen to the developing embryo. C. Gastrulation. In the third week of embryonic development, gastrulation occurs, which is the formation of three main germ layers. 1. Endoderm, the inner layer that will develop into internal organs such as the digestive tract, lungs, and thyroid gland. 2. Mesoderm, the middle layer that will form muscles, bones, heart, blood vessels, kidneys, and the reproductive system. 3. Ectoderm, the outer layer that will form the skin, nervous system, including the brain and spinal cord, and sensory organs. D. Neurulation. After gastrulation, the ectoderm layer will form the neural tube, which is the basis for the central nervous system, brain and spinal cord. This process is called neurulation. Formation of the neural tube. The ectoderm folds to form the neural tube, which then develops into the brain and spinal cord. E. Organogenesis. After the formation of the three germ layers and the neural tube, the embryo begins to enter the organogenesis phase, namely the formation of the body's major organs. Heart. One of the first organs to begin to develop is the heart, which begins to beat in the fourth week of development. Formation of the circulatory system. At the same time, the blood vessel system begins to form. Formation of other organs. Other organs such as the kidneys, liver, lungs, and reproductive organs begin to develop at this stage. F. Fetal phase. Fetalization. At the end of the second month, the embryo is called a fetus and the main organs have begun to form. Although not yet perfect, this phase lasts until birth and is marked by the growth and maturation of organs and body systems. Growth and maturation. Organs such as the lungs, kidneys, and heart begin to function more effectively, although the lungs are still not fully developed to breathe outside the womb. 3. Factors affecting embryo development. Embryonic development can be influenced by many factors, both internal and external. A. Genetic factors, chromosomes, changes or abnormalities in chromosomes, such as Down syndrome, trisomy 21, or Turner syndrome, can affect embryonic development and cause genetic disorders. Deep genetics. Some diseases or disorders, such as metabolic disorders or developmental disorders, can be inherited and influenced by genetics. B. Environmental factors. Teratogens. Environmental factors such as alcohol, drugs, viruses, or chemicals can damage embryonic development and cause birth defects or other abnormalities. Nutrition. Nutritional deficiencies, especially folic acid, can cause neural tube defects such as spina bifida. Infections. Some infections, such as rubella or cytomegalovirus, CMV, can cause abnormalities in fetal development. 4. The role of embryology in medicine. Embryology is very important in the medical field, both for diagnosing and understanding genetic disorders and for understanding the normal development of the human body. Here are some clinical applications of embryology. A. Diagnosis of birth defects. Understanding embryonic development helps in the diagnosis and understanding of birth defects or genetic disorders that occur due to disturbances in the process of cell division, differentiation, or organogenesis. Examples: Down syndrome, trisomy 21, congenital heart defects, or nervous system disorders such as anencephaly. B. Use in reproduction and in vitro fertilization, IVF. Embryology is also important in reproductive procedures, such as in vitro fertilization, IVF, which allows the fertilization of eggs outside the body to help couples facing fertility problems. During IVF, embryology techniques are used to select the healthiest embryos for implantation in the uterus. C. Cell therapy and regeneration. Embryology research also contributes to the fields of cell therapy and tissue engineering, by studying the potential of stem cells to replace or repair damaged tissue. D. Genetics and hereditary diseases. Understanding the genetic aspects of embryology is essential for the diagnosis and management of hereditary diseases and in prenatal genetic testing to detect genetic disorders in the fetus. Conclusion: embryology is the study of the early stages of development of organisms, from fertilization to birth. This process involves a series of highly organized steps, including cell division, cell differentiation, and the formation of the body's major organs. A thorough understanding of embryology is essential in medicine, as many disorders and diseases result from disturbances in embryonic development. In addition, embryology plays a role in reproductive technology and medical research, opening up potential in the fields of cell therapy and the treatment of genetic diseases.